Each registration is unique and grants the right for the individual registrant only to view this webinar. Sharing this webinar with unregistered viewers, either during or after its performance, violates Pharmatech Associates' copyright and is prohibited. Next. If you have not already done so, we ask that you take some time to read the Pharmatech Associates course disclaimer. You can download this presentation, including the disclaimer, from your USB Education account. Please follow the instructions in your confirmation email to download the presentation. Next. Welcome to today's USP Pharmatech webinar, From Experimental to Accepted, How In-Silico Modeling is Refining CMC with Regulatory Confidence. Thank you for taking your time from your busy schedule to join us. Presenting today, this webinar is Bikash Chatterjee, President and Chief Science Officer at Pharmatech, and Nima Jazdapana, Principal at Procigens. Welcome and thank you to our instructors. I will now hand it over to Mr. Chatterjee. You're muted.
There we go. Is that better? Can you hear me?
Yes.
Okay, perfect. So welcome everyone. Today we hope to take you through a glimpse of what is really happening in drug development today as it pertains to the application of in silico modeling. This is a evolution that's been coming for some time now and has reached a point where not only are the application tools at a point of maturity where industry can consistently and confidently implement them, but regulators also have a very keen idea of what the expectations around their implementation and are becoming more and more comfortable with the use of in silico as a core component of your CMC strategy. So our hope is at the end of the day today, you will walk away from this webinar with an understanding of some of the key ways that in silico is being applied and how regulators are looking at it. My colleague today, Nima Yazdampana, is going to be walking through some real-world applications of projects that have been utilized in silico modeling and have been successfully taken through the FDA and other healthcare authorities. Nima, you want to take us through this digital transformation sort of timeline?
Hello, everyone. Thanks, Bikash. So I'm excited to dive into the technical side and show you the real-world examples of the how the in silico modeling is making differences in the process development, the scale-up, tech transfer, and eventually to the regulatory submissions. As you can see here on these slides, so there are lots of new tools and the evolution of the computational models and the technologies that has been implemented in the pharmaceutical and also fine chemical and other industries that now is acceptable by regulatory agencies. And so different tools, different applications, and different methodologies that should be applied for different kind of steps of the process, from discovery to formulation to equipment characterization to process design and the process development, all the way to the process control and eventually for the regulatory submissions. So it's needed to mention that there's not one tool fit all. So we need to have like different approaches for different kind of technology and the steps of the process. For example, for like earlier stage formulation development, we have to use some like molecular dynamic modeling. Going to like equipment characterization, that would be some like a CFD or computational fluid dynamic modeling that would be used, and then going to like flow sheet modeling, and then to the process control or like the PAT or data analytics that's gonna be kind of integrated together. And at the end, it's gonna be like the holistic approach to combine all of these together based on the knowledge, based on the data, based on the information that gathered from material to equipment to process, and would have like the holistic approach to show sensitivity analysis, design space, better control, faster product to market, and the benefits for the process and also the patients.
Yeah, I think this has probably been the, Nima, your time at FDA when you were there in the EET.
Yes, yes.
ATT, I mean, this has really been the biggest evolution, I think, in thinking is that the agency now seems to have a much better understanding of what these tools can do and how they relate to classical experimentation and trial and error sort of drug development. Isn't that true?
Exactly. So at my time at FDA and the emerging technology team, so we work with different pharmaceutical companies, and FDA, as an example, is very kind of supportive of these kind of model-based process and product development. There are a couple of guidelines out there, and this is based on the quality by design, which is knowledge-based process and product development, and understanding the connectivity of different parameters and showing them by the models and the simulations that would be helpful for like risk analysis, process development, and control strategies development. My point was regulatory agencies, I was part of them before, and they are very, very supportive of this approach.
Right. So you're going to hear a lot of terminology in silico modeling. You hear this notion of digital twins, modeling, simulations. I think it's important to have a very solid grounding in what the different applications of in silico are. Maybe Nima, you can talk about a little bit very simply what the differences are between these three different terminologies.
Exactly. Thanks, Vikash. So this is not on a like a different specific hierarchy. I would say maybe the modeling would be like the fundamental of the other two. So developing some sort of equations or the correlation between properties and the parameters in kind of mathematical kind of description of the product or the process. Then we go to simulation, which is going to be running different models at the same time to kind of imitate the behavior of the process or the product, and then eventually go to like a digital twin, which is the kind of highest maturity of the combination of the simulations and the real-time data that could come from the process, and have some like a digital shadow or digital copy of the process and the product that we could use for many different applications, including advanced process control, real-time release testing, and then also kind of predictive maintenance.
Right.
Next
So when
slide, please.
Yeah, there you go.
This is an example of how things are working together. So it's just kind of a snapshot of one of the projects that we have had. You can see on the center. So we have the modeling section, which includes different kind of equations for material, for process, for equipment, for connectivity of all of these together. And that at this level, we can define what properties or process parameter or CQA is important for us, and we can develop some dashboards, and then combine with some like a statistical toolbox. Is going to be combination of the mechanistic modeling and the data-driven models that would go through the simulations and then eventually to the digital twin. Next slide, please.
Yeah, and so when we think about, go ahead. When we think about in silico modeling here, there are a lot of different tools that have matured here, but they really can be sort of categorized into three fundamental buckets here that most predominantly are being used, and then we have this sort of hybrid interconnection. We could talk about, Nima, a little bit about how we're using this today.
Sure. So mechanistic models are based on the first principle analysis. They are based on the thermodynamics, physics, kinetics, chemistry, all of these kind of physical and the first principle understanding of how things work together. The benefits of mechanistic models are like they are data agnostic, so we don't need to have a lot of like a process data and the equipment data for developing these mechanistic models. Very, very useful for a scale up and tech transfer. Either the DOE at manufacturing scale is going to be very expensive or impossible, or we don't have enough material to run at kind of larger scale. So that's we go with the mechanistic modeling. AI-driven or let's say data-driven models are more useful when we have enough good quality data that we can run and train these kind of the data-driven models based on existing data. Maybe for like earlier stage, if we have some let's say high throughput screening data that we can develop those kind of AI models based on the existing data. And eventually, these two categories of the model, they have their own benefits and the challenges or shortcomings, that we combine the good side of the both categories to have some sort of hybrid approach, which could be some parameter estimation that could come from the AI models, and then combined with the mechanistic models that could cover for different kind of application that you want to use for the process development.
Yeah, I think Nima, you touched on something that I want to make sure that everyone gets, this notion of mechanistic modeling as it relates to first principles. First principles sort of obviates the classical problem we've all heard about.
Correct.
AI hallucinations, over-constrained models that we have, which results with outputs that when you train a data set, it looks like it's analyzing the data, but it's actually just learning the behavior between the data set you have here. But mechanistic modeling bypasses sort of encumbrance, because data is always the challenge in pharma. While we think we're a data-rich industry, when it comes to AI, we don't have anywhere near the data typically we need to train. And so by relying on mechanistic modeling, we're able to basically get down to fundamentals of chemical engineering, physics, and the other behaviors which don't really care about the data you have. This has really been one of the biggest evolutions, and one of the reasons we've been able to see such tremendous acceleration when it comes to the CMC side, when it comes to scale-up process design and stability. We'll talk about some of those case studies.
Correct. So we're going to show during the case studies and then discuss about the scale-dependent versus scale-independent properties.
Yeah.
And as you mentioned, for tech transfer and scale-up, when we go to CMO with a different size of the equipment, how we can use this mechanistic model to predict the performance of the process and the product in a larger scale.
And this works with both small and large molecules. So
exactly.
So it has really been a powerful tool for accelerating a time to market. So as I mentioned, both Nima and I mentioned, FDA and EMA have been advocating for the adoption of these predictive models for more than a decade. I think in 2004 is when the first PAT guidance came out in the US, advocating for the adoption of chemometric modeling. The industry's been slow, quite honestly. Technology's been evolving slowly and getting better day by day, year by year, as has been the software, and more importantly, the understanding around how to apply these tools and what they mean in terms of their ability to predict accurately what's going to happen in a process. As we look at the situation today in a snapshot, the FDA recognizes in-silico modeling, as does the EMA. So if you wanted to sit down with the agency at a pre-IND meeting and tell them, hey, by the way, we're going to do our module 3 in silico before we go to experimentation, they would be very receptive to that conversation, to your plan there. EMA supports mechanistic simulations out there. They want to see the structured approach out there. They see the validity of that, partially because we can interrogate the data 100, 1,000, or even more times more deeply than you can experimentally. So what we'd normally do in 5 or 6 or 7 or 8 lots from an empirical perspective, we can now do 100 DOEs and interrogate the model that way and challenge ourselves running different simulations as to whether the conclusions and the predictability are robust or not. Both agencies accept integration of these in silico methods along with your conventional drug development on stability side, on process development side there. So I think you're going to find a receptive dialogue around this. They've set the context for what they expect you to develop as you're putting together these models, the notion of context of use as you step into designing these particular models. I think the expectation around the framework for using these models, around data quality, on how you do calibration and verification and validation are pretty well defined. So both industry and regulators are looking at the model from the same perspective. At least in the US, there are numerous pathways for interaction with agency around how you're going to develop these models. Regulators on all the major markets are talking about predictive modeling in a number of different applications. You've heard some of these other areas like toxicology, physiologically based pharmacokinetics. These things are more diagnostic and supportive in terms of how the agency looks at them. But when it comes to CMC, the ability to apply these models very effectively as a surrogate or a very strong complement to your normal development process design exercise is really well established. So it is sort of a bellwether for what the future is going to look like. As Nima mentioned to you in that slide, the application of In-Silico, there are a myriad of applications that can effectively leverage the power of In-Silico. Together, we've got more than 27 different use cases and applications, so highly tailorable. They're powerful tools and toolkits that can be used. This is just to illustrate to you some of the key target areas where the application of In-Silico could dramatically shrink down not only time to market, but also in today's financially challenged marketplace, it's highly capital efficient. And by capital efficient means it costs a lot less than empirical manufacturing of a batch, an experimental batch, in order to gather the data. So we're seeing very targeted applications here that allow us to develop high quality data, run simulations, and really quite at the end of the day, we're going to save significant time and significant money when it comes to the development of our module 3. So Nima, why don't you walk us through sort of 3 sort of real world case studies that sort of illustrate the power of how In-Silico can dramatically simplify and accelerate the classical elements of module 3.
Sure. Thank you so much. Next slide, please. So a couple of case studies that I've here for you. So talking about a process, a product, and also like equipment. Here is a spray drying process that we wanted to have a scale up from pilot plant to manufacturing. This is from mobile miner. Now we're going to PSD4. And you know, this equipment is like a 2-meter diameter by 4-meter height, so very kind of expensive process, and includes like many different scale-dependent properties and phenomena. For example, from atomization all the way to drying and the particle shrinkage, and then to the cyclone for the particle separation. So when we develop this kind of mechanistics model, it's very important to have multi-scale, multi-physics, and multi-domain model developments. So it's starting from atomization. You can see on the top right that we see what would be the impact of the viscosity, surface tension, density of the feed, which would be the feed composition. Different solvent, different excipient, different API would provide different CMA, critical material attribute, that would go to this atomization, and that forms different droplets that through the spray dryer chamber, they're gonna going under the heat, the mass transfer and the heat transfer, evaporation, and the particle shrinkage. Also, there would be some coalescence. Some of the droplets gonna hit each other and coalesce together, which all of these gonna impact the CQE of the product, which would be the particle size distribution and also the residual solvent content. So starting from like the very the first step of the process, which would be atomization, then we're developing this kind of knowledge about the droplet generation, the coalescence, the drying, the shrinkage, and what is the behavior inside the chamber, right? What is the residence time of the particles inside the chamber? How long they are exposed to high temperature? Is that high temperature gonna induce some impurity formation, which is gonna be degradation of the material, and eventually get into the CQE of the product.
I think
Go ahead.
Well, I was going to say, one of the things I really like about the application of In-Silico here is that we work with lots of drug sponsors all over the world that are attempting to put together their filings for the US here. And the notion of quality by design, in which you identify your CPPs and your CMAs, is one that still people struggle with out there. And so oftentimes it's hand-waving exercise as it moves from R&D to the shop floor out here. But by building these In-Silico models, you are forced to really drill down on what the CPPs are, and then you're able to demonstrate their impact on the final product CQAs. So it's giving you a complete QBD argument at the end of the day. So it de-risks your regulatory filing, because at the end of the day, the selection of your CPPs and the justification of them are obvious because they're fundamental to your in silico argument.
Correct. And we're going to see that in the next case studies, if you could proceed, please, that how we can use these digital sandboxes to develop the design space, run different process parameters. How about if you're going to higher temperature, higher RPM, lower pressure, and what will the impact on the CQA of the product? So this case study is for the bioreactor scale up, which also would be very similar to like the hydrogenation in a small molecule synthesis. Is a kind of same kind of physics that's happening there, but we have this kind of bioreactor scaling up from like, let's say 1 liter or 0.1 liter bioreactor going all the way to manufacturing to 10,000 to 20,000 liter bioreactors, and you can see the kind of non-linearities in the scale up. When we are going from lab scale to manufacturing, we cannot maintain the same RPM. If the lab is at like 150, 200, at the manufacturing, we might be at the same or even lower because of all the shaking and the vibration that happen at the larger scale. At the same time, the volume going to be like 200,000 times larger, and the VVM going to be like 100, right? So there is a lot of non-linearities that, you know, for the scale up, it should be studied and kind of figure out the correlation between the parameters. Next slide, please.
I just wanted to comment here that I think one of the things that we classically have done to try and obviate exactly what you just highlighted, Nima, is this notion of building a scale down model, because we are ultimately, for cost purposes, one, but also our ability to control. We'll do this at pilot scale. And at pilot scale, it becomes the basis for our CMC. But there's always risk just because of these nonlinearities as you scale the process up to 10,000 liters or 5,000 liters or 20,000 liters or beyond. We're doing a project right now where they want to go to over 50,000 liters at commercial scale. So
Correct.
Anyone that works in the large molecule space knows cells don't like to be under pressure. And so when you go to 20,000 liters, 50,000 liters, cells are under pressure. So how do we deal with these sort of the cell morphological components and realities of that, and find a design space that's going to allow you to optimally optimize that particular unit operation? And so this is something that we can run in silico and optimize, and you'll see in a minute how we do that.
Correct, yes. As I explained about the mechanistic modeling, now we're going to see how we're going to define the different interfacial properties and the phenomena at molecular scale to cell scale to the liquid and the process. And it's very important to understand this correlation from the first principle approach. For this kind of bubbling fermentation, that we have the gas bubbles that go up from the sparger all the way to the bioreactor. We have the gas liquid mass transfer at that kind of like the blue region that is you can see on the left side, and there are many different properties and the parameters that gonna impact the KLA. For example, the RPM of the mixer and the type of the impellers are very important for breaking down the gas bubbles. For example, if you have a Russian turbine, you're gonna hit the blade is gonna hit the gas bubbles and break them down. Now we have a smaller and more number of the bubbles that would increase the KLA. When we go higher level in the height of the bioreactor, now these bubbles are gonna coalesce together, and we have a smaller ratio of the surface to the bulk, and we get less KLA. Same for the like what's happening inside the liquid. Now we have the transfer of the oxygen from the gas surface to the cell surface. Now the media are going to be important: the density, viscosity, working volume. You know, many different things from the material to the process condition to the process design that's going to impact this kind of mass transfer. And then on the right side, we have the liquid to solid mass transfer, or KLS, that how would be the oxygen transfer rate to the cells, and when they consume the oxygen and make the product. Now we have the CO2 that goes back to the liquid and should be stripped off. Same for like the product, it's going to be monoclonal antibody or cells or proteins that's going to be released into the liquid. So defining all of these equations and kind of systems of the different models would be a step one. Then a step two, combining all of these equations together and run at the same time, because it's a process that everything happen at the same time simultaneously. That is called now to the simulation. So it goes going back to the previous couple of slides that I mentioned. You see the model developed. Now we are running all together. You can see this, the triangle of everything works together, which called the simulation now. And in next slide, you can see the workflow of how we go from like the model to simulation to digital twin. So this case study we had from the lab scale 0.1 liter to 20,000 liter manufacturing scale, and this kind of interesting the design concept that in the lab scale we have option, or we have the luxury of trying different reactor, different RPM, different impeller, because these are cheap and we can try different kind of equipment setup. But when we go to manufacturing, we cannot have like 5 different reactors and see which one would gonna work best for us. So there are some like capital intensive, let's say optimization, which we try to avoid. I don't want to buy a new bioreactor. I don't want to buy a new impeller. How I can use my existing bioreactor or my CMO's bioreactor to get the better results and more efficient results in a shorter time. So in this case study, we have 3 parameters that are gonna be almost free to change. For example, the RPM cost no cost. There is no cost for changing the RPM. So the orientation of the impellers, can I have like an elephant ear on the bottom, then rush the turbine, or kind of switch them around, and also the VVM, the gassing rate. These are 3 different, I would call it free parameters that we can change without having any capital cost. So these are like a constraint for optimization. Next slide, please. So for the workflow, so we can see how we get the data from the lab scales, what properties and the parameters that we need from the cell biology. This is going to be your cell, your chemistry, your molecule no better than anyone else. So we need to know the cell biology or the reaction kinetic and the behaviors and the limitation of the cell. For example, what is the sensitivity to a shear? If we go a higher RPM, because in more shear, are we going to cause some aggregation or the damage to the cell or rupturing the surface of the shells, right? Mm-hmm. Then from there we go to the development of the model, and we would have like the model validation and the verification, which is very important. Then we go to the manufacturing scale by these validated models. Now at this kind of manufacturing scale, because we have validated and verified models, we can use for like extrapolation, developing the kind of virtual DOE, trying different conditions, different properties, different parameters, and developing that design space that you are going to see in the next slide. And eventually from this kind of property estimation and kind of scouting different parameters, we get to the CMC module 3 for PAR and NOR and the design space. In this slide, you see the results from the case that I kind of explained in the previous slide, and we run different VVM, different RPM, different orientation of the baffles and the impellers, and then we can see like a theoretical design space that, hey, if I go to like, let's say 100 RPM, what would the impact on my KLA, right? So that theoretical design space might be like not efficient to implement in the process, which would be like costly or damaging to some to sell, or just maybe my equipment is not capable to transfer that kind of demand. That those boundaries gonna define our design space. For example, the green section here kind of exclude the conditions that gonna damage to the cell. The purple one gonna be like the costly experiments, and the blue one would be the starving for the cells. Now we can see we came into this design space on the middle that would be efficient, that would be implementable, and is designed based on these kind of in-silico models. Next slide, please.
Yeah, just one last comment here. So I hope you're getting an inkling now. The complexity that Nima is highlighting here. This is what we deal as process engineers with every single time we do attempt to scale up a large molecule process. The issue is our ability to see and to do the what if scenarios is very limited experimentally. So that's one problem. Secondly, every time we propose a PAR and a NOR in our CMC package going forward, there is a risk that the FDA is going to challenge that as we put our specifications in place. The ability to build this model in silico there and interrogate it and challenge it to come up with this very focused design space provides us the justification for whatever we end up with in terms of our final power and NOR. And our strategy around power and NOR, as you know, it can change as you're looking to implement your final control strategy. We now have exquisite sensitivity around what the implications are. If we move up, we move down, we shift left, we shift right. So this gives you all of the horsepower you need to have confidence in your final power NOR, and secondly, also justify what your limits are, which is compelling when you're putting that in front of the FDA or EMA.
Correct. Yes. So this is one dashboard from like my side on the like the modeling and simulation setup. I just wanted to show you with like a full transparency that how things work. So this is the dashboards of running different kind of case studies. On the top left, we can see every point or every line would be a new case. So now we are running 200 different 250 different combination of the VVM, RPM, orientation of the impellers, and we run it like very kind of quickly in silico. The lower left is the response factor. So if I change my RPM, what will on a KLA? If I change my orientation of the impeller, what will the impact on a shear rate? And now from that dashboard, we can get the responses and the risk analysis, and also use this for a reduced order model that's going to combine with some AI tools that could be used for the process development, risk analysis, and the design space that Bikash mentioned. Can see we can develop this dashboard very quickly and running this on the high performance computers. Now I'm changing to I'm running 250 experiments within few days, right? So it would be impossible to do it in the lab. Either the equipment doesn't exist or the material would be very costly, and also the time. Right? Next slide, please.
Yeah, I just want, and so I think this, hopefully you're beginning to get a sense now, if your manufacturing scheme and strategy includes an outsourcing strategy in which you've got a CDMO partner now that we're going to transfer this process to their particular plant.
Mm-hmm.
We've got to get into the queue, we've got to get into the schedule. We want to be as time and capital efficient as possible, so that when we build our lots, we use that lot to maximum advantage. We can do all the what ifing here.
Exactly.
And just demonstrate the predictive accuracy of the model with perhaps a clinical resupply or a PPQ lot or a registration lot, something we have to build anyways. But the likelihood of failure is incredibly low because we've already set the boundaries to a point where we're confident that we're going to be well within the design space.
And also comparing two different CMOs or two different equipments. So we could do this kind of what if analysis, that if I have this vessel with this geometry versus that vessel with the CMO2, which one would be better in terms of the yield, the cost, the time, and we can also have this kind of intelligent CMO selection with a lower cost and better performance.
Right.
So this is the kind of outcome of the model that I showed you. We developed many of these dashboards and the graphs. Here you see two examples on the shear rate versus KLA or the volumetric the kind of gassing time for different kind of combination of the process properties or the parameters that you want to set up, and that provides the holistic view of all the sensitivities, all the problems, all the benefits, all the that we call it kind of design space. Now the benefits would be, I'm kind of repeating myself, without spending any time in the lab, any wasting material, media, or cells, we can predict the performance of the process and the product and the equipment, and have some like the predictive recipe or SOP for how to run the process. Do I need to fill it up all the way to 100%, or maybe I can sacrifice some of my, let's say, RPM, and then I reduce my operation cost. So combining these together also get to the techno-economic analysis and balancing the OPEX versus CAPEX versus how should I run that, and eventually time and cost saving.
Right.
Next slide. Now we are moving to the third case studies, and every of these case studies it will be like the days of the discussion. So I will just very quickly jumping over a couple of the cases for different applications. Now here we are showing you kind of for the stability prediction for a oral solid dose, and there's been like significant time saving, and is already submitted. We have a couple of projects submitted to FDA and is supported by FDA. They have a product called MARS, M-A-R-S, which is predictive stability based on the degradation, based on the humidity absorption, any phase transformation, anything that would happen on the API within 24 months, 36 months, or 48 months, but by limited data and predictive models. In this example, you can see we have different kind of strength in the tablets, so we expose them to different temperature and humidity. We develop some kind of data that could be used for model validation or model calibration. Next slide, please. And then everything based on the first principle. For example, here is a kind of reaction kinetic that we developed for that would follow these. For this example, follows the Arrhenius type reaction kinetic, and now the temperature variation would be the activation energy. We have the concentration, or like Ca, which would be, let's say, humidity, and we can predict that at what temperature, what humidity we would have the degradation. So the mechanism would be different for biologics. So the protein aggregation, they follow different models and different equations, or inorganic versus organic small molecule would have different kind of equations. But we wanted to show you here that how the overall approach works and how we can develop these models to predict the stability for the 2 years or the 3 years based on limited data. And again, FDA is supporting this approach, and there have been a couple of workshops and the guidelines that developed that how to use this predictive stability to save time and then bring the product to patients faster.
Yeah, I think, folks, this is an area that has been a high baud rate of conversation with the FDA. We've participated in a number of workshops with them. Just the industry beginning to adapt ASAP testing, in addition to classical ICH stability testing, has been a long and arduous conversation, but now the FDA recognizes the value, at least for organic molecules, of ASAP testing here. Now the notion of bringing on a predictive model on top of that is they're comfortable with it. It's something that actually provides them with the confidence that in the absence of real-time data, that the product is going to behave as they expect it to go. And so this is a real accelerator for drug sponsors that are looking to buy some time, whether it's an ANDA and you're looking to accelerate that 6-month date, or a 505(b)(2) and you want to get that 12-month date down to 6 months, etc. This could be a significant time saver. Nima, you want to do this?
Yeah, this is just a summary of what we explained. Is also kind of tied back to the earlier slide that we have for like different modeling and simulation applications for different steps of the process and product development and along the life cycle. This is also not just for the process development, also we can use it for like CPV and the continuous process verification and maintaining the quality and the robustness of our process and the product. Again, I emphasize that we need to have different tools. There is no one tool that fits all. From the molecular level to the process level to the equipment level, we use different approaches specifically for that product or the process, the biologics or devices or a small molecule that can predict the process and the material and the product.
Yeah, so I think the toolkits have matured to a point now that we can mix and match. We can build hybrid models to answer these key questions that we typically, historically, had to try and do empirically with very, very little experimentation. So as you look at where we are today, this is just an example of classical process scale up, whether you're doing a large molecule or small molecule or complex large molecule or even API around it. The application, the targeted application of in-silico modeling can dramatically shrink the time it takes for you to get the answers that you need to put together a defensible filing here. On average, 60% time savings and cost savings is what we're seeing. Usually it's much, much larger than that. Obviously, if you're doing a biologic drug or a complex large biologic drug, those savings can be significantly larger. I just did a workshop in Germany at CPHI, and we talked about how all Well, most of the large biotech and pharma companies are formally integrating in silico as a precursor test to any experimentation. And so folks are beginning to understand and leverage the capabilities of in silico and the tools around them to allow us to do it right the first time. So we're taking the guesswork out, and that's really important when time to market and capital efficiency are front and center when it comes to drug development. So as we look at the CMC workflow here, it is really transforming quite rapidly and quite markedly. The notion of starting off with virtual experimentation, the ability to do scenario analysis, modeling, build a digital twin if necessary, understand the behavior based on very, I would say, lab scale characterization studies. This is flask level stuff that everybody does in R&D. As we move from to product design, we're doing this characterization work anyway. So by and large, we have the understanding that we need to build the models early in the overall drug development process here. So we're seeing virtual experimentation now in silico, allowing us to exploit that and take a lot of the risk out of the scale up and process design process, as well as the stability process, to argue that the product is going to be safe and stable and effective over the shelf life of the program. The regulatory dialogue is at a point where now both health authorities and drug sponsors are in a position where we're both talking from the same perspective. And that's taken a long time to get there, but the rules that have been put up by FDA and EMA make it easy for us as drug sponsors to put together a in silico model argument that's going to be defensible and compelling for regulators to review. Basically, regulators love this because it de-risks their overall evaluation exercise. We're providing them with 10-fold, 100-fold, 1,000-fold more data to answer the question that we're trying to answer. So it's a better situation for them. And that results in improved submission efficiency. Whether it's at the pre-IND level, we're going to advocate for this approach, or you're putting together an ANDA, and you can have a pre-ANDA meeting with FDA, for example, and tell them we're going to do in silico for our stability, we're going to do in silico for our scale up there, so that they're aware of it. They're going to love it by and large. BLA is obviously extremely expensive to do the scale up, and the engineering runs around that. We can do this all in silico today at a fraction of the cost, at a fraction of the time. So it really is a perfect convergence of both regulators, technology, and drug sponsors to be able to leverage this approach to be able to accelerate your overall CMC timeline while actually reducing regulatory risk. And I just want to end by saying that this little glimpse into some of the real world application reflect the broad number of projects that we have done together. So this has moved from theory to application, more than 20 large molecule projects, 60 small molecule projects. So the application of in silico as a core component of your overall filing is moving towards becoming the norm in that. And so I would encourage everyone, you can do this today because the tools have reached that maturity to be able to leverage their power in providing the answers to these core CMC questions we're trying to answer. So that's it for our presentation. We're happy to take some questions.
Thank you to Mr. Chatterjee and Mr. Yazdapana. Using the Conference.io software, please submit your questions through the social Q&A interface. You will also have the option to upvote the question by clicking the arrow next to the question. Next. Go back to back one slide.
Go back. I don't know if it'll let me go back. Let's see. Okay.
All right.
We have a couple of the slides in the chat.
Yeah.
Yeah.
I can read the question.
That's fine. Yeah.
That's fine. Yeah.
Yeah. First one, when presenting in silico modeling data to FDA as part of your CMC package, what's the one thing you learned that changed how you frame the narrative? What do they really want to see beyond the model outputs?
Can I take this?
Go ahead, Nima. Yeah, I'll chime in.
Yeah, from my experience sitting on both sides of the table. So when I was at FDA, someone would come and say, showing their DOE and said, I changed the temperature and this is my impurity. And we asked why, they had no idea, right? So they can show that, I test that, but I don't know why that happened. But with the models, so you're actually not going to show the model results. You are going to demonstrate your knowledge about the process and the product, right? So you're going to demonstrate that I understand what is sensitive in my process, why that impurity formation happened, why my yield is low, why this thing happened in my process, and how I can control that. So starting from just showing the data points to demonstrating your knowledge that I know what's happening there. This is my design space. This is my controller strategy. This is my risk analysis. It's going to be totally different type of language and the demonstration to the FDA. And FDA is been loving this approach over the past 5 years, at least the cases that I submitted to FDA over the past few years. And it also expedited the review time because they need to they could spend less time to review your application, and you're going to save time on the review cycle as well.
Yeah, that's a great answer. I think that is the heart and soul of what we're doing here in In-Silico, is that these are tools that provide insight as to what's happening in your process and what it means to your product. You still need to translate that to the regulatory understanding that you would typically put in a module 3 going forward. So the language isn't hugely different. It's the basis for the knowledge. It's like if you had a mechanism of action that you put together and you identified biomarkers. They're not going to focus on how you identified the biomarkers. They're going to focus on how the biomarker is indicative of the mechanism of action. That doesn't really change with In-Silico.
Correct.
Next question. What's the biggest gap between what In-Silico models can predict today versus what actually happens at commercial scale? How are you bringing that gap in your work?
So the purpose of building a model based on first principles is that the laws of physics are insensitive to the kinds of variables that we typically empirically worry about. And so when we build a digital model and run simulation based on the characteristics of your product, we've already incorporated the variables that you're typically going to encounter empirically. So in a small molecule, just to give you some level of variability, and Nima, chime in, you've got more data than me on this, but for a small molecule, typically these models run 5% or less in terms of their variation to actual. Large molecule, we might see as high as 20%, but oftentimes it's half that. So the beauty is that science is science and physics is physics. And by building the model for first principles here, we basically obviate the empirical nature of data, the impact it can have on your overall prediction capability, so that it's going to be in line with what you're ultimately going to see commercially. So the delta is very small.
Correct. Yeah. You can think about, for example, aerospace industry, right? So the Cassini satellite launched 20 years ago, right? 15 years ago, right? And they designed it based on the first principle model. So nobody could, send us like a shuttle to moon 10 times back and forth and then run a DOE, right? And that satellite went past the Mercury with the expected accuracy that they wanted to have. So it's but we have to draw a line between must to have versus nice to have, right? How much is the level of the accuracy you want to have from the model versus the time and the cost of developing the model, and what is the context of use, COU, that Bikash mentioned in the regulatory expectation when we see the models. Then we get to a lot of the scale dependent versus scale independent phenomena in the process. For example, reaction kinetic is a scale independent. It depends on the temperature and the concentration. But dissipation of the heat, mass transfer, KLA, they are scale dependent. So the scale dependent could be run and validated in a smaller scale in the lab. Then when we have the verified model, now we can go to like any larger scale and we can predict that what with the impact of mixing, the phase transformation, the let's say the immiscible fluid, catalyst, whatever we have there, that it could be predicted. But again, we come back and say, hey, I want to see, for example, I need to have like a diffusivity or let's say viscosity. Do I have it? Maybe not. Is it very important for the model? Maybe yes. That comes from the sensitivity analysis that we do at the model development stage and say, hey, viscosity is very important for us. We have to spend more time to predict that or by measuring it in the lab.
Yeah, in the lab.
Yeah.
Next question. For process changes post-approval, where do you draw the line between what in silico modeling can justify as a minor change versus what still requires new clinical data? How have you seen FDA's appetite for this evolved?
So I could start, and Nima, you could chime in.
Yes.
I think the ICH-Q12 has given drug sponsors a very clear pathway to deal with post-approval changes. And so we encourage drug sponsors as they're moving towards submission to put in place a post-approval change management plan in which we can talk about these kinds of changes. So if you plan to increase your process from, I'm making this up, 10,000 liters to 25,000 liter batch size post-approval on this, and you want to use an in-silico model to define the design space, et cetera, we would include that in our change management plan prior to submission, have the conversation around it, talk about what we're going to do and what we're going to present so that it becomes a very seamless and simple exercise. The argument around clinical requirement really is almost indication dependent these days, and perhaps mechanism of action dependent on a case-by-case basis there. So I think you have to plead that case early on if you do plan to make some significant changes to your overall formulation or your product or your process going forward. But the change management plan is what we use to put that in front of the agency prior to approving the first submission.
I agree. And
go ahead.
No, go ahead.
Next, what are applications of in silico to botanical drug development? That is, growing plants, extracting bioactives from plants, and developing final product with multiple APIs.
I could take that, or you can chime in, Nima, either way.
Sure. Yeah.
Yeah, so it's a multimodal problem there. There are core components, as Nima mentioned. If there are aspects of the botanical product that are essential to the final claims of efficacy, I assume this is an RX product that you're talking about, that you want to use as basically the botanical product is now the API, the drug substance. In that, we need to, again, characterize the key unit operations that are going to ultimately drive the final product CQAs, just like we would do normally in any sort of process development. The considerations around it are the same as you would have in a biologic or even a small molecule product. We need to identify which parameters are critical to the overall process, any material attributes, and then link them to the CQAs, just like we do in any control strategy. So I wouldn't attack it any differently. We've worked with a couple of botanical products. There are multiple botanical products together there, so it is a complicated undertaking. I will say that botanical drugs in the US are their own regulatory process because there are just some things around it which you cannot control to the same level as you would an API or a biologic drug out there. And so understanding what those differences are a priori before you build your in silico model is important to do. So I would encourage you to link that regulatory thinking and understanding up front before you build your in silico characterization work.
I can add more to the like the process development or like a kind of a scouting of different solvents or like the extraction as an example that we had a project 2 years ago. When we're extracting from leaves different kind of gums and the sticky material that got out. So now someone need to do the solvent screening. I'm using like hexane or ethanol or supercritical CO2. That cost because the material like very sticky, running in like a bottom run flask, then just 20%, 30% just sticks in the wall and not getting out, right? So for that project, so we help them with like a solvent selection and also then the purification. So yeah, they had to go to like a below the level of the solvent in the final product. That like a scanning different type of solvents and the process and the kind of process condition that they can run to get to the product that they want. So increasing the yield, how you can get the last the drops of the terpene you want to get from the leaves all the way to the quality of the product. So that could be like not much into the submission to the regulatory, but more for like a process development and the product development, huge time saving and cost saving.
The challenge you have with botanical drugs is the growth and harvesting and control of that at the different centers there. And so whether it's done in the US, I think the botanical drugs have all been done under highly controlled conditions so pesticide free. We understand the soil, the environmental conditions around it are extremely well controlled. You're kind of handling it like a normal biologic or a small molecule you would. Outside the US, there are botanical drugs that have been approved there, and they are, they're harvested and they are combined and characterized to some level, but obviously the variability can be quite large with that. And that's a source of variability you have to eliminate in order to put together an in silico model, which is going to be representative. Otherwise, the variables just continue to move, and that's a problem.
Correct.
How can modeling support equipment independent tech transfer, for example, in continuous direct compression applications?
Do you want to take that one, Nima?
Sure. So again, depend on the CMA, CPPA, CQA correlation, right? So the what is important for example, the solution and the drug release is going to be the type of the powder that goes to this different kind of compaction, or it would be the compaction force, right? So starting from the kind of developing that kind of knowledge map, similar to that the KLA that between the bubble to the liquid to the surface that I showed in the slide, developing from that level that I have a spray dried particles versus I have crystalline particles that would have different kind of need different compaction force. And if I go to this new dye for the compaction, what will be the impact of the pressure that gives the different tablet strength that it goes to the different drug release or the time profile. So is a nonlinear kind of answer. So it depends on kind of equipment, material, and the product. But it's very easy to develop these things. And at that point, I'm expecting the product owner to the kind of enough maturity level that have enough data. Now we can also have some sort of hybrid model, some mechanistic model about like a mechanical strength of the tablets versus some dissolution profile that came from the experimental data that we can combine these and have a holistic approach.
To me, the continuous is probably one of the best illustrations of the application of the power of in silico, because you have the potential for transient excursions in that, and you can predict it, you can simulate it, you can find a design space, and you can establish criteria for your CMAs and your processing conditions that will keep you comfortably within your 3 sigma variability.
Exactly. And also some experiments going to be very expensive. For example, RTD, if you want to do RTD in a continuous drug compression, so you have to run it for a longer time for different magnitude and the duration of the spikes to see where is my off-spec material coming, what's the impact on the solution time or the tablet quality. If you read the Q13 Annex 2 for the drug product, which I had the privilege to contribute to the Q13, so it mentioned about different models and different kind of simulations that should be developed to predict the equipment characterization, equipment performance, and also the RTD for rejection or for like the acceptance criteria.